degree in computer science from the University of Castellón in Spain. He has worked at EBI for about 1.5 years and is currently a software engineer working in the Intact group. Today he will present about PSICQUIC, which is a web service that standardizes the access to molecular interaction databases. Without further ado, allow me to introduce Oscar.
Okay. Thank you, Vincent. Well, I hope everybody can see my screen.
Yeah, looks good.
Okay. Well, hello everyone. My name is Oscar Forner. As Vincent introduced me, I'm from the EBI. I'm going to introduce you to the Intact project and PSICQUIC. The introduction to Intact project will be a brief one. It's just to let you know in what kind of databases is PSICQUIC relying. It will be a brief one. The presentation is mainly focused on PSICQUIC. Okay, first of all, what is Intact? Intact is a free open source database, and we also provide analysis tool for molecular interaction data, such as PSICQUIC. All the interactions in Intact are manually created. That makes us a high confidence resource. But what means manually created? Well, that's our Intact curation pipeline. Sorry. Everything begins with the publications. We have here the first step is the publication. If we recall from the previous slide, the interactions in the Intact database are derived from the literature creation or direct user submissions. As we can see here, the curator can do a curation manual or use an import. To do the import, it has to be in the right format and everything, but usually if the authors can provide it, it's easier than have to curate manually the publication. Once that is done, we have an entry that it can be annotated with all the information that the curator thinks that it's necessary or important to provide as well for the database. Then the next step is, once it's annotated, it has to be checked for another curator. In that case, we are going to call it super curator. It can happen two things. It can be accepted or rejected. If it's accepted, it will be publicly available in the next release of the database. But if it's rejected, it will have to go to the previous state to do for the original curator to do all the changes that are necessary. I don't know, maybe the super curator didn't like the way that it was annotated some parts of the publication or some experiments, and they have to be reviewed, and afterwards it can begin again the process of checking and trying again to be accepted or not. Well, and the last step is once the publication has been accepted, in our database we have several sanity checks and a monthly release of data. The data will be available in our FTP site, in the website, and other resources such as PSICQUIC. Now we are going to talk what is that resource called PSICQUIC. What is PSICQUIC? PSICQUIC is a way to standardize the access to molecular interactions databases programmatically from the HUPO proteomic standard interactive. Well, maybe this is not quite clear what this is PSICQUIC and how it works, but you will have a better idea in the next slide. Okay, that is a better view of what is PSICQUIC. Okay, this is the main infrastructure of PSICQUIC. We have here the services. All of them are provided for different databases. Intact will have one of them, but there are several other databases from molecular interactions that provide PSICQUIC services. We have as well the PSICQUIC Registry, the MIQL, that is the language that we will use to query, and the PSICQUIC View. Well, that's the big picture. Now we are going to go deeper and see what is each part of it. Okay, the Molecular Interactions Query Language, MIQL. It's a consensus between different databases, so you should be able to use the same query across different repositories. Okay, that was one of the main aims of PSICQUIC, because as everybody knows, you can't query in the same way in one database or in the other one. Everyone has their own ways to do it, to build the queries, to filters, everything. That was with the MIQL, we wanted to change that and has a standardized way to do it in all the resources. Well, it's based on Lucine's syntax. If you have ever used Apache Solr, Elasticsearch, or something like that, or as a search engine, you will be familiar with the syntax. It's not really easy to get it. It's quite simple. We have something called terms. Those are single words or phrases. If it's a phrase, it has to be quoted. If not, it will be counted as an operand, but we will see that later. For example, we have the BRCA2 or pull down quoted to just ask for that explicitly. We have as well what is called fields, used to search in a specific column. Example, species human. That means that we just want all the interactions that has human as the species. We have as well term modifiers that allow us to do wildcard searches, fuzzy searches, proximity, and range searches. For example, the BRC star, we can get the results from the BRCA2 because it's part of the end of using this wildcard. We have as well the operands. By default, it's an OR. If you don't specify one, it will be an OR. We have the OR, AND, NOT, plus, and minus. Plus is another way to write AND, and minus is another way to write NOT. In the example, we have the BRCA2 and RPA1. That means that we have two sets of results. Only the intersection of both will get it, the BRCA2, RPA1. That's really easy. Everybody that knows how the Booleans work, it's easy to follow it. And the grouping and field grouping, BRC2 and mouse in vitro. It's not a hard one, but it's more tricky than the previous one. We'll have to, first of all, resolve the parentheses as in math, and afterwards you will apply the AND. It's just to have more restrictive searches or more advanced ones, but they can be built easily once you get it how it works. Okay, well, that was the language that we use, but I was talking as well before about the registry. That's the registry. All the PSICQUIC resources has to be here in the registry. Right now we have 3,824,489 interactions from 31 different PSICQUIC services. Okay, what are those services? We have here, for example, we have several fields to describe them. That's the name. Usually it's the name, that's the name of the PSICQUIC service. And usually the name of the PSICQUIC service is the same one as the database that it's providing it. Okay, we have here the name, we have as well the status. Those are up, and that one at the time I took the screenshot was down. That is useful to see, for example, that when one is down, the interactions that are they doesn't count for that big account, that big count. But for example, for BioGrid we have 47,000 interactions. Well, anyway, here in the version we have the version of the PSICQUIC service that it's using it. For the general using that is from the PSICQUIC View, we'll see what is that. it's not really important, the version, because all of them provide the same functionality for the PSICQUIC View. But if you want to use some specific functionality or features that are from a specific version, you will have to query specifically that resource. And for that, we are providing two ways of querying it, using a REST or a SOAP. You have there two links for each of the entries and an example of the REST. As well, we have the restricted. As you can see, all of them are no. That means that they are freely accessed. We don't have to identify ourselves or anything, it's just to query. And the tags as well. The tags are important if we are going to filter which one of the resources we are interested in. For example, we can see here the protein, if it's imported, cluster, spoke expansion, it's referring that how they build a complex using just binary interaction. We have as well small molecule protein. That all depends on what kind of search do you want to do. You can just filter for the tags and see which are the resources that are providing the information that you need. Well, that's the registry, but that's not the most important one. The PSICQUIC View, that's the important one and the main way of our users to use PSICQUIC. Okay, here we can see all the services that we have right now to query. As we can see, by default, we are going to query all of them, but if we just want to query some of them, we've seen in the registry that we just want to query 3 of them because they have a specific tag that provides some information that we are interested in. We just can select them here and we will just query them. Okay, we have a legend here to see what are the state of the service. At the moment, all of them are offline or online. Well, here we have the search box. We are going to build our query. For the example, I'm going to use just the PIF3. That will be a simple one that we will be all able to follow. Okay, we search for PIF3 and in all the resources, and we got 223 binary interactions. But for example, if you check that, we got none one from Reactom, because Reactom don't have information about those ones. But just above that one, we have Mentha with 98 results. We have 98 binary interactions from Mentha. Or we have 51 from Intac. Okay, yeah, we know which one, how many they have, but how do we check them? How do we see them? Okay, if we go to the link of one of the resources and we click on it, it will show above in the bottom of the page. It will be shown those interactions. The interactions are shown in the MyTab format. That's a result format of PSICQUIC. It's a really easy format. For example, we can see here at the beginning that we have the identifier from the participant A, from the interactor A, the ID for the interactor B, the aliases for the interactor A, aliases for interactor B, a species from molecule A, species for molecule B, those are the interactors, the first author of the publication, the ID of the publication, the interaction type, the interaction detection method, all the information. If we are interested in just to see few of the columns that there are, we can just use those options, show and hide, and you can decide which one do you want to see. Or you can even download the file in several formats. But yeah, that's a way to represent the information in a table. But as well, we can show it in a graph using the Cytoscape plugin. We can see here how it looks, our search that we got in Intac. But yeah, we are just watching what's the result in one of the resources, but the good stuff is to see what are in all of them at the same time and see how they overlap and which one are duplicated in some of them. Okay, for that we have that button here called Cluster this query. If you press it, it will take a few seconds, depend on the complexity of the query and the amount of binary interactions that it will have to cluster. When I mean cluster, when I say cluster, I mean to try to find all the duplicate interactions and just get rid of one of them. Just will show one per. We have two interactions that are equal. We are not going to show both. We are just going to show one of them. Once it's complete, it's already clustered, we can just press View and we'll see the clustered interactions. You remember we had, as a result before, 223 interactions, and now we just have 51, but those are clustered. As I was talking, for example, we can see that one, the participant 080536 and the participant P14713. That one is a quite common one in all the resources that we got, and we have a lot of different authors, different publications, different interaction types, and different detection methods. Okay, that's the result of all our resources, and deleting all the duplicates. That's the important one. Actually, we got just a quarter of the original amount of interactions from all of them. But as well, we can see again in a graph what. Okay, that graph it has more information than it has in the previous one just from Intag. You can see from Intag we just had the PAF3 at the center and some of the links from here, but not all of them. Now we have plenty. We have more information here as well, and some isolated ones here. Okay, well, we are using Cytoscape graph again. And well, okay, that is PSICQUIC. Do you have any questions or something?
Thank you, Oscar. If you have any questions, you can voice them aloud or type them into the chat box.
Yeah, sure, I'll be available in the chat box anyway.
Hi, this is Vincent. So I posted in the message box, but I have a technical question. So how does one of the web services in the registry become compatible with MIQL?
Okay, that's not in the way that it's done. We provide some sort of a skeleton of a web service that you have to fill your data in. If you are familiar with, for example, Solar, have you ever worked with Solar for search engine?
I've heard of it.
Okay, the first step is you provide the data and it has to be indexed to afterwards allowed to do the queries. Then we provide all that information. We provide the Solar that it will have to use, all the Java and all the Java libraries to query it, and everything. It just the database or the database people, they just have to provide to map their information in our that format that is MyTab, and afterwards once they have built their own version of of PSICQUIC, because you just have to fill the skeleton. Once it's done, they just provide us the URL. We check that everything is fine. They can really answer the MIQL queries and everything, and we add them to the registry.
Okay. That makes sense. Thank you.
You're welcome.
So this is Andrew Sue. I have a question here. So what do you know about the use cases for people who use your, this interface or the web services?
Yeah. We have different use cases actually, because some people are, for example, interested in just the annotations that are for human, and they just want to see, for example, they know that in Reactom they have information, the pathways from human mainly, but they want to know as well what other people has to say about those pathways or those interaction that they found interesting in Reactom. And an easy way to do it, you just paste it here in PSICQUIC and you get the result, and you can compare anyway with what you get in Reactom. That can be one of them, but it's plenty of use cases depending on what you need. You just have to search for the right services.
But Reactom, right, is part of, right, is a PSICQUIC endpoint.
Yeah, they provide a PSICQUIC service, yeah.
Well, let me maybe ask a different question. I mean, do you find that people have, I guess, resource-specific queries? Like, is there a scenario where I would want to use, I don't know, innatedDB but not MINT?
Oh, yeah. Yeah, for example, there is a clear one that it's called Go, that they are from Go annotation. They provide just annotation for different interactions or different proteins or, information, but it's just annotation that they made from that information. If you just want to know, they are, how to say that, they are collecting all the annotations because, for example, some of the Mensa, they can think that that annotation is not really important or useful for them because they are focused in another research. Then they just keep some of the annotations, for example, UniProt, they can provide you more information in the annotations of the sequence or peptides or whatever they need. They are so different. But for as I was saying, that database Go, they provide all the annotations. They just get all of them and provide them. And they are right now they are planning to get a PSICQUIC service. Then, yeah, if you search for if you're looking for an annotation, yeah, the best way is to go there and ask them which one, what annotations they have for a specific protein or something. Then, yeah, the best is sometimes it's more than query all of them, just query few of them or maybe a couple and see what they have in difference, because I don't know, as I had to the example, the PAF3, we just had in Intact 51 interactions, but in Mensa they had it around 98 if I remember well. Okay, but maybe you want to know which one from them are repeated or which one we have duplicate if we compare with them. Then you can do those kind of queries easily with PSICQUIC.
Okay. And then what about sort of, understanding that there's a need to sort of compare, then what about the bigger picture use cases? I mean, do people come in here, what I guess are the input sources of the genes of interest? I mean, I assume, whatever, RNA-seq or a GWAS study or variant detection, and people are just trying to tease apart sort of networks in the protein interaction, or subnetworks of the protein interaction network that appear to be enriched. Is that basically it?
Yeah, that's the idea.
But then to do that, right? So for example, so let's see. Do you allow lists of genes or proteins to be input into the PSICQUIC view?
Yeah, you can do it. You will get the output, all the the interactions that has those genes or proteins are involved there or are related with them. Then, for example, how to explain that? Maybe in an interaction, we did for say something in reactant data is created for, because I don't know, in the paper that they were creating, they created just a few interactions but they get focused on one of them, and the other one didn't have all the information that someone wanted. But in INTAG or in MENTHA or in another one, they do it in the other way around. Then if you go to just one of them, you won't get the whole picture of the interactions that are related there or the reactions. Then when you cluster them and put them together, you will get what is really happening there. That was what we wanted to do with PSICQUIC.
But then I mean, really tease apart enriched subnetworks. I mean, you guys don't do any of those statistics within.
No. No.
Okay. Cool. Thank you.
You're welcome.
Are there any other questions for Oscar? Okay, thank you, Oscar, for that great presentation.
Thank you very much to everyone.
All right, now for our second presentation. Our second presentation will be presented by Salvador Martinez Bartolome. He holds a computer science degree in engineering from the Autonomous University of Madrid and a master's degree in bioinformatics and computational biology, and also a PhD in bioinformatics. He has worked in John Yates Lab at TSRI for a little over a year, and today he will be presenting about PINT, or Proteomics Integrator, which is a tool that serves to store lab results that can be consulted and reused, even if the producer of the data has left the lab. So without further ado, allow me to introduce Salvador.
Thank you very much, Vincent. Good morning, good afternoon to everyone. I'm going to present PINT, the Proteomics Integrator. So that is a tool that we have developed in our lab. In principle, was motivated by some situation that we think that most of the proteomics lab can have. The researchers generate some data, some mass spectrometry data, and then they do some data analysis pipelines, like the typical ones, to use a search engine to validate the IDs, make some quant analysis and some statistics. So can be some quite complicated data analysis depending on the lab or the goal of the project. But then this can be done, this pipeline can be done by some known softwares, but also it's typical to have some extra analysis by the researcher, like merging data from different assays, use of other software or scripts that are not available or are developed in the own lab, or so also to apply some custom statistics, like some custom p-value calculation or extra filtering to focus on some kind of some subset of the data, or even, for example, searching for protein annotations of the proteins obtained in the analysis. And typically these analyses are manual tasks. So at the end, we obtain some final results, and becomes somehow a problem because we want to handle all this data that comes from this extra analysis. We want to store the data, and also we of course we want to share the data. We want to publish the data and to share the data to the scientific community, and also for example, in some labs like this one, we have a lot of people that is coming and then they leave and with collaboration or something, and it's difficult to recover the data generated by other people that leaves the lab, and so we want a system to store the data and to provide the data to the scientific community. So the main motivations is to provide a long-term storage of the data of the lab, so don't let the data die when the scientist is gone, to share the data, as I said, and also to enable a sort of high-level analysis of the data using available protein annotations that are, in this case, in UniProt. So we developed this tool that is a web-based tool for storing and sharing the final proteomics results of the lab. So it becomes a local repository. So I want to clarify that this is not a substitution of the available public repositories. This is a local repository for the lab that can be useful for several things that I will explain, but is not to replace the public repositories like Protein Exchange or some of them. So to do that, the tool relies on a flexible data model in order to support complex experimental designs, like having multiple runs, replicates, experimental conditions, etc., and different quantitative approaches, different types of proteomics data, like different cell expression data, but also cross-links data or protein interactomes. And one important thing is that it's capable to import proteomics results even after manual or non-standard user interventions. So, as I said, use of in-house tools or manual analysis or additional filtering, etc. So then it's the ideal tool to manage and share the amount of data generated by a proteomics lab, and provides a high-level analysis of the datasets by a sophisticated query system that includes the UniProt annotations and the experimental setup of the data. So this is the schematic overview of PINT. So we have an input with some input data files and the experimental design, so the user has to provide this to the system, and then there is a sort of minimum semantic validation of the data made by, let's say, a PINT import interpreter, which say that if all the minimal information for store the data in the database is there or not, and if that data makes sense or not. So if it is valid, then the data is all integrated in the database, and also the system try to map the information of the proteins to UniProt. So sometimes the user provides the UniProt identifiers, but for example, we have a lot of data using the IPI database, so we have to map it to UniProt and then use the external resources of UniProt in the internet to get all the annotations of these proteins and also store it in our system, and then to be able to use these annotations of the proteins stored. Then the system is going to check if a new release of UniProt is available, and then it's going to retrieve the annotations of the new version of UniProt. This is made because sometimes an experiment is done like several years ago, and we want to use the annotations of the data that were available at that time when the data was generated. So then PINT provides the possibility of use old versions of UniProts from the date where the experiment was deposited in the system. And then PINT also provide a query data system, so the user provide a text with the query over the data, and this query is parsed by the system, and on the data of the experiment is retrieved, and also the annotations to build a query result for the user in the interface. So the data model of the tool has to be flexible enough to support different levels of data aggregation. So we want to take into account several levels of the data, like several projects, experiments, replicates, runs, but also the actual data at PCM level, peptide level, protein level, but also the protein groups, meaning so we perform a protein grouping with the proteins that share peptides. We also want to, as I said, to support different type of quantitative approaches and also cross-link data. Regarding the data query system, is a command line-like system, but assisted by a web GUI. Currently, we have plans to improve this GUI in order to make even easier to send the commands to the system. The queries or the commands are based on MS features, so the features about the MS data, but also, as I said, can be based on the protein annotations in UniProt. But also, the user can provide, when import the data in the system, can provide manual annotations on the data, but also custom thresholds or scores, like for example in a proteome interactome, the user can define some thresholds in which the proteins that belongs to the core interactome or are below certain threshold. So this can also be provided to the system and then can be used in the queries. And in that sense, the queries are not only queries but also are filters of the data to visualize in the interface. So this is how the interface looks like. There are a sort of query commands and can be selected, and then the user can write the query on that. As I said, we have plans to improve this interface and will be much more intuitive for the user to write down the queries. A brief explanation of the available commands right now. So, for example, this one is they are like this with the key of the query and then some parameters. So in this case, the cont command is used to select the data from a certain experimental condition in a certain project. So in this example is to select the protein detected in SAHA condition in the project, called project whatever. Another query command can be based on a certain score condition. So in this example, for example, we want to select the PSM detected by an associated score of type PTM localization score. I have to say that these names of scores and are mapped to the ontologies on used on or defined in the PSI, the Protein Standard Initiative. So these terms can be used in a lot of fields in the commands. So then the query is to select the PSM detected by associated score of type PTM's localization score name site localization confident equals to high and above a fault discovery rate threshold of at petail level of less than 1%, for example. This another command is based on the ratios and the amounts. So, for example, proteins with a quantitative ratio measured between condition SAHA and condition control with a value greater than or equal to 2 and an associated p-value less or equal than 0.001. So as you can see, you can build quite complex queries over the data, and this can be very useful to make high-level analysis or inspection of the data that we have. Also, as I said, you can select the data with certain UniProt annotations. We provide a lot of keywords of the annotations. And also, we have right now also commands for select the data with certain isotopic labelings or certain taxonomies or gene names, etc. So these are the commands right now, but as long as the user are using in the lab right now the tool, we are adding commands very easily. So I think that the list of commands will grow over the time. And then we can combine several commands like in the MIQL that was presented before. So we can use the logical operations and the not, for example, here, to make complex queries like this one. For example, this query means give me the proteins detected in condition disease A that are not present in condition disease B, containing an associated p-value less than 0.05 and with a twofold upregulated from control, and that having 7 transmembrane regions annotated in UniProt. Okay. Then how to import the data? So we have built a kind of import data wizard. So the idea is to import the data from different sources. So we now support output from our own analysis tools that we use here, like DTSLET and Census. These tools are included in IP2 software suite that was presented the other day by Robin Park, but also we want to import the data from any Excel file. So how to do that? So the system should interpret the input data. So the user has to upload the input files, the system has to recognize them, and then the user defines the experimental setup, the experimental design, so the project, the experimental condition, the runs, etc. And then the system somehow will ask the user, where can I find the proteins, the peptides, the score, the thresholds, the PTMs, if they are available in the Excel file, which files are related to each condition or run, for example, between which conditions are the ratios calculated, etc. So this is done in this interface. So at the left we have this blue box in which the experimental setup is defined. So the user will choose how many experimental conditions, how many ratios between two conditions have, how many samples, MS runs, how many data sources, which means how many input files or input files is going to provide. These input files can be located in remote servers, so the user can define server connections with using SS file connection, which is the sample origin, which means the tissues or of the samples, the organisms, and also the labels. So then there are some boxes in the interface which are going to provide the user the way to relate the experimental conditions. For example, in this box, each one to which sample is going to be related, the ratios between which conditions are going to be calculated, the sample, each sample, which label has each sample, which sample origin, which organism, etc. Other examples, the MS runs, the files, the data sources can be the Excel file that you can upload, or that, as I said, can be located in some server, and etc. Then, once the experimental setup is defined here, then the user will have to define, for example, if it is using an Excel file, it will have to select the Excel file, and then, because this is an example of how to define a ratio, so the system is going to detect every column in the Excel file, assuming that has some header in the first row, and then the user will have to select the column, and once the column is selected, it will show the values in the Excel file to provide a way to the user that that is the column that he wanted to select, and so on. So in this case, the user is selecting one column to define the ratio between KO and wild type, and also another column to define an associated p-value of that ratios. Okay. So then this is our import quiz data wizard, and then we have the visualization interface that is based on Google Web Toolkit library, and is able to visualize the data at protein level, PCM level, but also protein groups. This is how it looks like. So we have here a set of options for the visualization, and then several tabs with the proteins and peptides, etc. The typical interface of proteomics data. So in this case, you can see that for each selected protein, we see here all the peptides, and also the quantitative data with the amounts and the ratios between conditions. Okay. The user can also select a lot of columns that are available for proteins, for PSMs, or also protein groups. The user can select several projects if they are available to query or to visualize the data on all of them. Okay. And one thing is that the protein grouping that we do is based on the protein inference algorithm called p-analyzer that was published 3 years ago. And so the data can, if the user select the protein groups view, is able to see each protein group and the type of the protein group in the interface. So to finalize this presentation, I will show you some future developments. So we want to provide a wider support for input files, so probably to use the PSI standards like MZ-Tab, MZ-IdentML, or MZ-QuantML. That is something that we have to consider, because in this lab we are not using these standards right now, but we want to do that. So this is one thing. Then we want also to include cross-link data support. That is something that will take some not too much effort for us. We have some data also on that, so we want to include that type of data in the system. As I said, also we will provide new query filters commands. So right now the system is very easy to implement new queries commands. So will be the list of commands will be longer in the time. And we want also to link to other tools like PSICQUIC, a protein set enrichment analysis that we have developed in this lab also. So we want to, as long as it is possible, to link our data to third-party tools to use them as more easy as we can. So. And we can also provide a query, the query system as a web service interface for third-party tools in order to provide other tools a way of querying over our data with all the commands that we have. That is something that we have not done yet, but is something that we'll do. Conclusions of this talk is so the PINT is a tool for storing complex proteomics results. So it's a local repository. It's not intended to replace any public repositories. Focus on a local repository for a lab or a set of labs, but is able to import proteomics results even after manual or not standard user interventions. You see, we consider that is an ideal tool to manage and share the huge amount of data generated by the proteomics laboratory, and it provides a sophisticated query system that provides a way of high-level analysis of the datasets. Well, that's all. That is our PINT server, where it's located now. The Java code that is open source is located here in Google Code, and that's all. So thank you very much. If you have some questions, I can try to answer.
Thank you, Salva.
No questions?
So this is Andrew C. here. So pretty cool system. So I assume it's your goal to get this used in labs beyond the Yates Lab. Any comment on traction there and how that's going?
On any comment on what? Sorry.
I mean, I assume you want PINT to be adopted by other labs.
Yeah.
Has it been?
No, not yet. Not yet. In fact, it is working right now in our lab at some. As long as the users, the users here are starting to use that some few months ago. So we are still improving with their feedback. So as far as I know, there is nobody that is using that. We have not published this system yet. So when we feel that the system is ready to be published, we will publish it, and we hope that can be used for in other labs. I mean, that's true that it's true that I have been contacted from several people to want to try it, but as far as I know, they don't have it still installed in their systems. Yeah.
Got it. Thanks.
Hi, this is Sarah Scruggs. I just had a quick question. I was kind of in and out. There were some other things going on. So you may have mentioned this. But at what different levels do you store the data? I know this is kind of a big issue because space is always an issue. But so I know the raw file and then probably the sequest or census search results file, right?
Mm-hmm.
And then what other sort of levels do you store the data at?
So you mean which kind of data we import? So yeah, we import right now we can use the DTSLED IDs. So the IDs from DTSLED that can come in our lab is used over sequence and produce results, and also the quantitative data from census. But as long as a user has a table, any data can be uploaded to the system. Yeah, but yeah, mainly IDs and quantitative data, but also with several manual annotation thresholds, etc. As I commented. Yeah.
Okay. Okay. It's very cool. Nice work.
Thank you.
Are there any questions, any other questions for Salva? Okay, thank you, Salva. Great presentation.
Thank you.
If anyone has any other questions, we can continue the discussion either afterwards or next week. But yeah, thank you for participating in this conference. Just a quick reminder, so I sent out an email regarding our usage of GitHub for this project. Please read the email, look over the guide, and email me if you have any questions. Other than that, that's it for today's conference.
Was your question in the chat box, was that answered?
Oh, that was from the previous.
From the previous?
Yeah.
Okay, so it was answered. Okay.
Yeah. Yeah. If there aren't any other questions, then we'll conclude the conference here. Thank you for participating.
Thank you, everyone.
Thanks.